Okay, so we are beginning chapter 18 here, the renal system. Our first learning objective, so we're going to discuss all four of these learning objectives, which will take us into renal balance, which are the next upcoming packets. But we're going to begin with objective one here. So this is to describe how the urinary secretion of solutes and water influences the volume and composition of plasma, and then identify how other processes that can also affect plasma and volume composition. So really getting into the basics of renal physiology, and looking at the glomerulus and talking about filtration. So we'll talk about the functions of the urinary system, the basic renal exchange process, again, starting at that glomerulus. We'll then move on to look at the renal tubules. So the different parts, the regional specialization of the renal tubules that lead to different types of reabsorption and secretion. And then finally, kind of the ending process to create urine, which is the excretion. So let's recall some of the many functions of the urinary system. So the kidneys are two small organs. They're in the back of your abdominal wall or your abdominal region, close to the posterior abdominal wall. And they seem very small and unsuspecting, but they have a lot of different functions in many different body systems. So they can help regulate plasma ion concentration and composition. They can help to regulate plasma volume, so the amount of fluid circulating in your plasma, thereby affecting things like your mean arterial pressure and your blood pressure. Regulate your plasma osmolarity, so the amount of sodium and other solutes in your plasma, again, affecting the volume. And then they can also regulate plasma pH. So we'll talk a little bit about the acid-base balancing and the acid-base function of the kidneys. This is also an important aspect that happens with the lungs. So both the kidneys and the lungs together can help us regulate our pH, our body pH. And so we'll look at how the kidneys play a role in that and sort of touch on the lung function as well. Now, the kidneys can also remove metabolic waste products. This is the whole purpose of urine excretion, is to eliminate those byproducts that have been filtered from the plasma, so elimination of waste. Other associated functions include secretion of erythropoietin, which is the precursor to generating new erythrocytes. They can also create renin. Renin helps us to balance our plasma levels as well. And then hormone secretion. So vitamin D3 can be converted or activated into its active form, calcitriol, as well as gluconeogenesis, which is a part of our energy metabolism balance. So really important to think about its sort of plasma-related functions, which is what we're going to focus mainly on, especially during this session. But it also has some other functions that are not necessarily plasma, like hormones, energy metabolism, and erythrocyte production. So let's recall here some of the functions associated with these structures of the kidneys. So we talked about the area like the renal corpuscle. This is involving the glomerulus, and the glomerulus is really that ball of capillaries that's stuffed inside the Bowman's capsule, this outer capsular structure. The glomerulus has an afferent arteriole. This is the blood vessel coming from the main vessel or the main artery that supplies this part of the kidney or the glomerulus. And then it has an efferent arteriole, which is leaving that glomerulus as well. So the afferent is going towards the glomerulus. The efferent is going back towards the venous system. But before it gets back to the venous system, it's actually going to be very closely associated with the rest of the renal tubule. So this yellow structure underneath, which is the distal part of the renal tubule, right? So we have the Bowman's capsule, the proximal convoluted tubule, the descending loop of Henle, ascending loop of Henle, and this region that dips down into the medulla is called the vasa recta. very small capillaries coming from the efferent arterioles that are tightly associated with the loops of Henle. Then we have a ascending loop of Henle and a distal convoluted tubule that comes back to connect to the glomerulus. Really important to look at this relationship. The way that the distal convoluted tubule comes in very close contact with the kind of the opening of the Bowman's capsule. This creates a structure that we'll talk about, the juxtaglomerular complex. Very important relationship here. Then we have the distal collecting tubules, and then that leads into the collecting ducts that eventually merges to form the ureter. So these are the structures that we'll be speaking about. We want to remember their relationship. Again, this juxtaglomerular complex connection is going to be important, but also just thinking about how the initial site of filtration, we have that plasma being squeezed across the glomerulus, and then the filtrate accepting things like large proteins, erythrocytes, they're going to stay behind in the capillaries, but the rest of that plasma is going to get squeezed across to create our urine filtrate initially, and then that will be fine-tuned as we move through the rest of the tubules. So starting with the proximal tubule, loop of Henle, distal tubule, all of these locations of the tubules are going to have their own role to play in fine-tuning that filtrate to create our ultimate urine, which eventually gets released through the ureters and then stored in the bladder. So let's begin to think about that exchange process. First off, we want to describe our glomerular filtration. This is the process of squeezing blood across the glomerulus and into the Bowman's capsule. And so that is where we have the initial exchange where the plasma from our blood vessels can be filtered across the structure in order to create, I'm going to call it our filtrate for now. It will become urine, but it's going to go through a lot of conditioning. First off, it will go through reabsorption, where we can reclaim some of those things that we didn't want to secrete or to excrete, and we can pull those back into these peritubular capillaries. We're also going to have secretion. Secretion is in the opposite direction. It's pulling things that didn't immediately get filtered out, still in the plasma, but we want to secrete them into the filtrate so that they can be excreted. So these two processes are going to be dependent upon our body state, right? If we're hydrated, dehydrated, the amount of ions, sodium, potassium that's in our blood, certain other hormone levels like renin, angiotensin. So a lot of different factors will determine how much of different types of substances are we reabsorbing and how much of those other substances do we need to secrete to be eliminated in the urine. And then the ultimate elimination process is our excretion. So from here on, we send that urine out into the bladder via the ureters, where they can eventually become or be eliminated through the urethra. So just to recap that process here, our afferent arteriole bringing that blood in, our glomerulus, our ball of capillaries that are stuffed inside the Bowman's capsule, our efferent arterioles. So not all of the blood that ends up in the glomerulus gets filtered across into the Bowman's capsule. The rest of that flows back towards the efferent arteriole, and then it eventually ends up in the peritubular capillaries that are going to be intertwined with different parts of the tubules in order for these two processes to take place. And then after we have the fine tuning of our filtered plasma, then we can eliminate or excrete that as urine. So let's go through these processes one by one, starting with filtration. So this is movement of protein-free plasma from the glomerulus across into the Bowman's capsule. So notice this is not going to include proteins. When we look at the structure of the glomerulus, although it has the fenestrations, these endothelial cells have really large openings in between them. This is called a fenestrated type of endothelium. The openings are not large enough for proteins to escape. So proteins will not be in our Bowman space or will not be found in our filtrate. This is why protein should not be in your urine. If you begin to have spilling of protein into your urine, that is a disease state called nephrosis. And that can be a symptom of other diseases um, that we'll get into. But there should not be any urine filtered across your glomerulus or ending up into your Bowman's capsule. The other thing that should not end up there are erythrocytes. So again, there shouldn't be any blood in your urine or red blood cells in your urine. So those two things are going to stay behind. The rest of that plasma will get squeezed across the glomerular filtration membrane or the glomerular barrier. Now, there are three structures that the filtrate must cross to go from the capillaries and end up in the Bowman's capsule. This includes the fenestrated capillaries. So here in the lumen of our glomerulus, right? If you think about this ball of capillaries stuffed inside the capsule, if you look at the lumen of those capillaries, again, they have fenestrations because they are fenestrated capillaries. So they're fine-tuned, they're suited for this type of filtration of a large amount of plasma. They also have a basement membrane, this small, thin purple membrane, which is in between the two other layers of our filtration membrane. You can see that better down here. And then on the other side of our basement membrane is the visceral layer of the epithelial cells that create the podocytes of our Bowman's capsule. So there's the visceral layer of our Bowman's capsule, and that layer is not continuous. Instead, the cells here are divided by these filtration slits. So this creates these podocytes as well. And podocytes just mean foot processes where the cells kind of stick up, these epithelial cells kind of stick up. And then in between the membrane of these cells, we have these slits, these filtration slits that allow that plasma to be squeezed across into the Bowman space. So this is the visceral layer of that, and that is continuous with the parietal layer, which is out here in the Bowman's capsule. All right, so blood is essentially squeezed across these three structures, across the fenestrated capillary, of the glomerulus, across the basement membrane separating the two, and then across the filtration slits or the split pores of the other side of the Bowman's capsule, right? These are epithelial cells on the other side. So endothelial cell, basement membrane, epithelial cell. This is very reminiscent of our respiratory membrane. Very similar mechanism of our respiratory membrane, which means that this type of layer is suited for exchange. It's suited for the filtration and movement of plasma across these structures. Looking at this a little bit more closely, so again, our ball of capillaries covered with this visceral layer of the glomerular capsule or the Bowman's capsule, and that is reflected to become the parietal layer, which is out here. So that creates a space, visceral layer, parietal layer. In between those two layers is our filtration space. And so that is where our urine is going to, our filtrate at this point is going to initially be created, and then it can move its way into the proximal part of the tubule and start making its way into the tubular structures. And then what has not been filtered will actually move its way into the opposite direction back into the efferent arterioles towards the peritubular capillaries. Looking more closely at these membranes, we can see the filtration, the fenestrations of the capillary endothelium, which is suited for this type of exchange. We can see that thin glomerular basement membrane separating the endothelial layer from the epithelial layer. And then we can see the podocyte foot processes, which are separated with these filtration slits. This is where that filtrate will squeeze out and make its way into the Bowman space. All right. And then looking at what this looks like on a microstructure view, so a histology view here, again, our filtration slits, our foot processes sort of sticking out in between those slits, our basement membrane in between the epithelial layer on this side, the endothelial layer on this side, and then the fenestrations in between the endothelial layers of the glomerular capillaries. On this side would be the capillary lumen. Let's keep going. Now we're going to introduce the Starling forces. This is something we have also talked about before. So this should all be coming back and be a refresher in terms of these forces. We're just going to apply them to a different part of the body as they apply to filtration here. But the same general principles will apply. So our hydrostatic pressure, it's a push force. Our glomerular hydrostatic pressure is the push force exerted by the blood that is present in the glomerular capillaries. So that is going to be having an outward direction from the glomerular capillaries into the Bowman's capsule. Then we have our Bowman's capsule oncotic pressure. This we describe as a pull force. And this is also going to be in the perspective of the Bowman's capsule or Bowman's capsule over here. And then it's pulling fluid from the glomerular capillaries. So it's in the same direction as our glomerular capillary hydrostatic pressure, but it is created from the Bowman space instead. Now, interestingly, there should be no proteins in the Bowman space. And so our Bowman's capsule oncotic pressure should be nearly zero. And only if we have disease states, as I mentioned, such as protein spilling with nephrotic syndrome or any other type of glomerular basement membrane damage or injury, then we're going to find protein ending up in this space, and we may see a higher Bowman's capsule oncotic pressure. But normally, this should be zero, very similar to our interstitial fluid oncotic pressure. Next, we have Bowman's capsule hydrostatic pressure. So another push force, this time it's from the perspective of the Bowman's capsule or the Bowman space. So this is the push force exerted by the fluid that is in this very small confined space. And that is pushing in the opposite direction back into the glomerular capillary. Finally, we have the glomerular capillary oncotic pressure. This is again a pull force. This time it's from the perspective of the Bowman, of the glomerulus. So the fluid here, there's also an abundance of proteins. Because we are filtering plasma, we're losing some of that volume, but we're not able to filter the proteins. We now have a proportionally higher amount of proteins in a smaller amount of fluid. So our glomerular capillary oncotic pressure is usually relatively high when compared to other areas of the capillary. All right, so our hydrostatic pressures are push forces from either perspective, either the capillary or the Bowman space. And then our oncotic or osmotic pressures are going to be pull forces, again, either from the perspective of the capillary here or the Bowman space. And later on, we're going to plug in some of these values and actually calculate the glomerular filtration pressure. But for now, we're just introducing these forces, and then we'll plug those values in and talk about how we can calculate that coming up. Let's pause for a few questions. So thinking about the direction of the movement that our Starling forces are favoring, and which ones are going to be in favor of filtration. So first off, our glomerular capillary hydrostatic pressure. We know that that is a push force. It's from the perspective of the glomerulus, and it's pushing out into the Bowman's capsule. So this is going to be favoring filtration. This is the direction that we want our plasma to move in order to be filtered across that membrane. The Pressure here is usually 60 millimeters of mercury, and it's going to be high due to the resistance of the efferent arteriole. If we look at the lumen size of our two arterioles that are making up the glomerulus. So here's our afferent. It has a bigger lumen, it's a larger vessel. And then here's our efferent with a smaller lumen. So that creates this force that kind of traps pressure within the glomerulus because we have more flow coming out here, but less flow in this efferent direction. And so that allows for more of that hydrostatic force, that filtration force, to push plasma across our filtration membrane. The other force that's favoring filtration is our Bowman capsule oncotic pressure from the opposite perspective, but this is favoring pull or, pulling, which is also in the direction of filtration. However, this is going to be very low, again, zero in normal healthy individuals or normal healthy kidneys, because there should be no oncotic force. In other words, no proteins in our Bowman's capsule to exert that pull force from the glomerulus. So our overall forces favoring filtration should be about 60 millimeters of mercury only. If we look at our opposite forces that are kind of going against filtration, these are going to be the other two. So Bowman's capsule hydrostatic pressure, this is the push force exerted from the standpoint of the Bowman's capsule. And this is about 15 millimeters of mercury. Again, we have a large amount of volume in a very confined space in our Bowman space between those two layers, the visceral layer and the parietal layer of our Bowman's capsule. So a small space, high volume creates a relatively high pressure when compared to other areas of our capillaries. So this is about 15 millimeters of mercury. And then our other pressure, which is again much higher here, is our glomerular oncotic pressure. This is also going against filtration. It's the oncotic pressure in the glomerulus that is owed to the higher amount of plasma proteins in a smaller amount of volume of plasma. Just reiterating this here again. So because we have filtered some fluid across, some of that plasma has moved across into our Bowman space, we have a smaller amount of plasma and the proteins can't go. So if you look at these green, large solutes here on this side, these are representing proteins. They cannot go across that membrane. And so they're going to have a higher concentration in a smaller volume of plasma, which gives us a relatively higher glomerular oncotic pressure when compared to other capillaries. So this is about 29 millimeters of mercury, right? So if we kind of add those up, that's going to give us a combined force opposing filtration of about 44 or so millimeters of mercury, right? We're going to add our 29 and then add that to 15. That's about 44 millimeters of mercury. Ready? Let's keep going. So just to reiterate that here, so our Bowman's capsule hydrostatic and our glomerular capillary oncotic, those are both opposing filtration. So that is 15 millimeters of mercury plus 29 millimeters of mercury. And then the forces favoring filtration are the opposite. Our glomerular capillary hydrostatic going in that direction, and our Bowman's capsule oncotic going in that direction as well. Remember, this guy should also be zero because there should be no proteins here. And then that should give us about 60 millimeters of mercury. And then if we subtract that 44 from that 60, that should give us an overall glomerular filtration pressure of 16 millimeters of mercury. And our bigger force being the filtration force, being the overall direction of movement of that plasma. Okay, so really important to think about how the same forces, the same equation, the same principles of our starlings that we talked about with our interstitial tissue and our interstitial fluid, and those principles can be applied here as well to the glomerulus and the filtration membrane. Okay. So this is just a little schematic that I drew just to illustrate those, right? Our efferent arteriole, this should be smaller, afferent arteriole larger, creating a higher um, hydrostatic force in our glomerular capillaries. And so this is part of the reason that we have that big filtration force because this pressure is really, really high because of that smaller efferent, larger afferent, and creating a high force to allow for that filtration. So our overall calculation here should bring us back to that 16 millimeters of mercury. And so if you're having a difficult time following this, definitely try to draw it out. Draw the arrow so you can see the direction of the forces. Put in the values, plug them in. Overall filtration minus overall opposing filtration, and then you can work it down to your overall value. You should be comfortable doing something like this for the exam. So definitely go back over this if you're having a difficult time following this. And